we spend a lot of time in academia, but at the same time, we want to have that like Harvard meets MTV vibe where like people are excited. How do we make stem cells cool for kids? I know it sounds weird, but these are the questions that like we sit around and talk about. I'm Ben, I'm the co-founder and CEO of Colossal.
I'm Matt James. I'm the Chief Animal Officer at Colossal.
A couple years ago, we decided that in addition to the work that we are doing, we started funding about 7 to 10 million bucks a year in labs all over the world for ancient DNA research. Like we funded projects like ancestral models for like migratory patterns of cave hyenas, right? Just not that we're ever going to work on cave hyenas, but we just found the research interesting, right? And we thought, and we work with so many incredible collaborators that spend their time on our project. We will always try to support their projects too. And one of our great advisors from day one, who's a top evolutionary biologist, Michael Hofreiter from the University of Potsdam, literally since before we launched the company, was like, I know we're doing the mammoth. I know you want to do the dodo eventually, but we have to do the bluebuck. And we supported his lab and others, and we ended up getting a really good genome a couple years ago. And then, we stood up the Colossal Foundation. We started doing a lot of other things. And some of our partners came to us from a conservation perspective and said, hey, do you have any technologies that could be applied more to the bovid tree and more focused on antelope conservation? And so it was kind of a perfect storm, right? We had a great genome that we supported. We have a scientific advisor that's like absolutely in love with this. It is a terrible story, kind of like the dodo and the thylacine, where human, drove this animal to extinction. And so it's like, it started to check a lot of boxes, right? Like we had the DNA, there's a close enough relative, people in South Africa that we talked to were really excited about it. And, there's a huge opportunity for us to, then support those and develop technologies that can then be applied to antelope conservation. So it was kind of like this perfect like alignment. And so we started on it a couple years ago, and we were debating on when we were going to announce it. But if as you get to know us, you'll learn that we have in our de-extinction kind of stack, we've got everything from the ancient DNA to the cell culture to the editing to then the assisted reproductive technologies to creating stem cells. And like 4 out of the 5 like major things that we have to do on every project, we got done on the Bluebuck. So we're like in the editing phase. And so we decided, hey, maybe we should just tell people, we've made a lot of progress on it, and hopefully that also opens the door for using some of the tech that's already been developed for antelopes into conservation faster. Our goal is, we're in the editing phase, right? And so we've created the stem cells in their own. We've done a lot of the assisted reproductive technology work on kind of the back end surrogacy side. We have our targets from our computational analysis of the genome, and we're editing. Not only does it help us like develop a whole new skill tree, like apply our technologies to bovids, which can open up a lot more really interesting things, including commercial applications with like, livestock and others. And so it's a really interesting pathway into further developing the tech down that path. But also, one of the edits that we're making, there is most of the work that we do is around multiplex editing multiple parts of the genome at the same time, putting in different pieces, genomes, knocking pieces out, and then changing nucleotides. But what's interesting is in the geographic region of one of the changes that we want to make that's definitively Bluebuck was it's a very large cargo delivery, and it's the largest thing that's ever been delivered into a genome. And so that kind of said, we said, okay, well, we'd use multiplex editing and keep editing this, or, there's no like rush on the project. So why don't we develop the DNA? Why don't we further our DNA synthesis tools in our large cargo deliveries? Because then that's just another tool in our overarching tool belt.
Ben had described some amazing reasons why we're working on this project, and the plan is definitely to make a living, breathing bluebuck, and the plan is then to take that and put it back into the wild in South Africa, right? So we're working with amazing people at the Endangered Wildlife Trust as a conservation partner to help us begin to rehabilitate ecosystems and find places for the bluebuck to thrive again. What's really intriguing, I think, about this bluebuck project is that the bluebuck was really sort of the first large mammal to go extinct after like the whole Linnaean period. So after Carl Linnaeus created the whole modern taxonomy system that we know today, it was really the first species to be scientifically described as going extinct. So it was taxonomically described. 34 years later, it went extinct. So there is sort of this moral responsibility that we carry to help restore this amazing species.
Most people don't know around 30% of antelope species are endangered with extinction and threatened with extinction. And in talking with a lot of our conservation partners through our foundation and others, we found that about 75, there are kind of 75 partners, we found that it is one group that's massively underserved and underfunded for that reason. Like, I think that their impact on the ecosystem and universal love, but I think that because they are typically categorized in documentaries and film in reasonably large herds on camera, I'd say the general knowledge that they are threatened with extinction. I think people think that it's something just not to worry about. And that's some of the feedback that we've gotten from different conservation partners that focus on antelope across Africa is like, hey, raising money for things like rhinos, it has been significantly easier than raising money to support antelope conservation. And so we think it's also a really unique opportunity for us to bring awareness to that. It's not a zero-sum game, right? And I think that's a scarcity mindset versus abundance mindset, right? And thinking about like technology. And so all of the technology that we make on the path to bringing back a species is open source for conservation. And in addition to that, in addition to the money that we raised for Colossal, we raised $100 million for our foundation, and we're supporting other people developing new technologies as long as they open source it for free for conservation. And so we look at, so these de-extinction projects are not, we're not going out to like, the governments and foundations and NGOs to give us money and taking away money from conservation. This is new money into conservation from that's invested through a company that's developing technologies that, while it can be leveraged for conservation, can also be used for human healthcare, defense, industrial use cases, and others, right? And so we look at it as like we can subsidize developing technologies for conservation at a very, very rapid rate, while also bringing awareness to these problems, while also and getting kids excited about science and aware of the problem versus just the doom and gloom we hear on the news all day, right? It's like, oh, you can innovate and build new technologies to fix some of these problems, right? And so we look at it as, one, it's new technologies for free. Two, we look at it as it's completely new money into conservation, right? So this is adding new money to the pot of conservation, not taking away from conservation. And three, it's bringing awareness to these problems in a way that like will get attention.
Ben's spot on. The big piece of this is conservation, and our fight against biodiversity loss is grossly underfunded and under-resourced. We need to begin to innovate in the space and begin to develop these technologies. That starts with having new ideas, new goals that act as this engine of innovation and this engine of funding and awareness that Ben described in a way that can help sort of, lift all ships together. By investing directly into technologies that support de-extinction, we're creating technological platforms that are saving species today. I mean, you look at what we're doing with red wolves because of the dire wolf. You look at what we're doing for elephants because of the mammoth. Same thing because of the bluebuck. We are creating new reproductive technologies that allow us to begin to help address things like genetic rescue in critically endangered antelope species, address things like fragmented populations that lack gene drift between the populations.
This is not an alternative to conservation. This is just a new tool, right? It's a new branch of conservation, right? And so our view is that, you still need to protect land, you still need laws to protect species, you still need all of the things that we've been doing. You need to continue to do. But what we also know is what we are doing is slowing the process. It's not stopping the process. And so like we need new innovations, we need new technologies. And to Matt's point, it's like if the current types of work is underfunded, then you're not going to be able to spend that money developing new tools and new technologies, right? Because there's a cost in R&D. And so if we can come in and develop new tools and technologies for free for the industry and for everyone in conservation, we look at that as once again, it's not a zero-sum game. From a process perspective, it's very, very similar to all our mammalian workflow, but different than our avian workflow. So we still had to go build a reference genome. We had to do a complete assembly across the 5 genomes we were able to get from an ancient DNA perspective. We had to do all the comparative genomics. We had to identify those targets with this large cargo thing that we're working on. Probably be 100 plus edits in the Bluebuck versus 20 in the direwolf and 8 in the mice, right? And so we're continually advancing that. Just so you have an idea of scale, our mammoth project, we're doing about 150 to 200 edits at a time right now. So we're scaling up the multiplex editing. Once again, we're taking on this project, we are working on a couple of different new technologies, just because we think it'd be interesting. We think it's interesting. We don't necessarily need them, but for this project, but they're interesting long term for us. So we don't have a Bluebuck today, but this is something that we'll see, I think, in a couple of years, not in like 5 or 10 years. It's a 9-month gestation, so it's nearly a year of gestation, but we're in the editing process. And so, I think it's highly likely in the next year or two, we'll put embryos in, and then 9 months later, hopefully we'll have Bluebuck.
One of the key technologies that we've had breakthroughs with is a technology called ovum pickup, and that's the ability to aspirate oocytes out of an ovary of a live animal. So we've completed a novel pathway basically to do that in roan antelope. That's working really well right now. So we're collecting oocytes. We're doing all the embryological pathways right now to do in vitro maturation, in vitro culture. Eventually we'll start with IVF, so in vitro fertilization, which is what we've just achieved. So we do have embryos of roan, but now it's edited. Now we're waiting for those edits, so then we can do somatic cell nuclear transfer of edited cell lines. So that's sort of the next step. Ovum pickup technology is this amazing idea that we can use an ultrasound, a special ultrasound probe, and a special needle that has this that has suction and fluid within it. And you actually it's almost like a microsurgery where you use the ultrasound to guide a needle into the roan's ovary, exactly into an emerging follicle. And then you can flush fluid into it and then suck the fluid out of it, and it comes with the comes with the oocytes out of that ovary. So it is a revolutionary technology for things like antelope, because it gives us this ability to now leverage great reproductive technologies like in vitro fertilization, somatic cell nuclear transfer, in ways that we can help improve things like gene flow between highly fragmented populations. From a computational standpoint, it makes it more challenging for us to identify what are the Bluebuck specific genes. But from a genetic diversity standpoint, one of the beauties of the ways that we've developed our de-extinction pathway here is that we're actually capturing a lot of the background genetic diversity of the host species that we work with. So the roan antelope come with plenty of genetic diversity. We can edit on top of that in multiple individuals to achieve Bluebucks who have a nice diversity within the population. Our goal is always to create interbreedable, sustainable, self-sustaining populations. Like I described with the background genetic diversity that's achieved by editing on top of a roan platform, we're able to actually take genetically unique roan, edit the Bluebuck specific regions into each of those, and they still get all the background diversity that ensures that they have the nice healthy diversity within the population. So we should not experience a genetic inbreeding depression.
We edit multiple cell lines, and so we don't use just one roan genetic donor. We establish multiple cell lines from multiple different roan from different locations, so that baseline genetics is different. They could possibly say it's not the Bluebuck, but it is a definitely Bluebuck. So I don't think they can argue on this one. So we'll see. And so that's one. And then two, if we're a big freedom of speech company, and so if people want to call our mammoths, great. If they want to call them cold adapted Asian elephants with edited mammoth alleles across 104 mammoth reference genomes, through 1.3 million years of genetic drift and divergence, that's cool too, right? It's like, we that's what you want to do, like that's what you do. But it's like you cannot clone from a non-living cell. And so like our mammoth project is a great example. We have 104 mammoth genomes, over 1.3 million years of genetic divergence. Assuming, which we don't, assuming that if we had mammoth cells from each one of them, and I could clone one of them, which one's a mammoth, right? And what I can tell you is it's not the sum of the parts, it is the parts, right? And so over 1.3 million years of genetic divergence, there are parts of the genome and genes that are conserved that truly make that a mammoth and classified as a mammoth. But there is a wide range of genetic divergence. And so, when people want to have a taxonomy or philosophical argument about it, we just take the opportunity to educate them. And, if they, agree, great. If they don't, great. I don't think we have enough going on. It drives me crazy. And so, I mean, building and scaling companies has been my background for a long time, right? And you have to build teams and trust them, right? And so for us, we have 260 plus scientists alone, then support staff, 300 plus employees at Colossal. Then we have 17 academic partners. Then we have 75 conservation partners, right? Because we don't think everything on conservation has to be invented here. We've got 80 postdocs in academic labs, and we have 95 scientific advisors. So, I mean, that's a lot of people. It's 500 plus people. That's not a, it's not 3 postdocs in a lab, with prayer, right? This is, we are, and we're well capitalized. We just raised more capital earlier this year. We're now doing work with government, so some of that will fuel even further expansion. But these are individual groups, right? And so we have the core. We run the company less like a biotech company and definitely not like an academic lab. It's ran more like a product company, right? And so for us, we've got our core platform teams. We have our individual species teams. We have our support teams. We have a foundation which is completely separate, right, on the conservation side. And then when we spin out and license technologies, that has new teams and new capital that goes with it, right? And so we, to Matt's point, I don't think we're doing enough.
And I just think that we do feel an immense sense of responsibility. I think there's so much out there in the natural world that needs healing, and we think we are capable of achieving amazing things. And so you have this sense of responsibility that you want to be able to say yes to as much as possible. You want to be able to save as many things as possible. We're lucky because Ben has done a great job of helping develop this business into something that's able to attract a lot of funding in ways that no other conservation organization in the world has done. So we're fortunate because with that responsibility, we also have opportunity. So we're going to continue hammering away on as many of these projects as we can.
But it's not, to your point, it's not a 9-to-5 job. Like you come here and, there are people working at night, people working on the weekends. People are doing this because they are truly, truly passionate. And there is a sense of urgency. People aren't just like meandering through the hall. Like there is an excitement and energy here, which is exciting. One is continuing to make all the core edits that have been identified from the reference genomes. And then 2, really focusing on seeing if we can get this, we haven't done it yet. So, I mean, I don't know, but if not, we'll just go back to we'll just edit it, not the old fashioned way, but our old fashioned way. But the next step is on that side is like, we have the DNA synthesis and we've got pretty close to done. And then we have a way of delivering it into the cell, and it's, DNA is fragile, right? So that's complicated. And so if that works, then that'll be great for just a new technology tree for Colossal that's interesting around DNA synthesis and large cargo delivery, or I should say larger cargo delivery. And so those are probably the biggest next steps. And then once we get to a point where we've made all the core target edits, we'll do the somatic cell nuclear transfer into egg cells, and then hand off to Matt, the science team hands off to animal ops team to put them into our own.